Hello, my name is Patrick Kenney. I'm a graduate research assistant at the University of Missouri, and today I'll talk to you about how to manage sour rot of grapes. In this presentation, I'll cover what sour rot is, the causal organisms, and then go over management strategies for this disease. So what is sour rot? Sour rot is a late season bunch rot. It typically develops on tight clustered thin-skinned grape varieties, so this could be red or white. And berries show discoloration starting about 2 to 4 weeks before harvest, and sometimes in Missouri, even earlier than that. There's a wound site that is required, which could be separation from the pedicel or berry splitting, as well as insect wounding. And there's a strong smell of acetic acid or vinegar within the vineyard. This also is associated with a loss of berry integrity as the pulp liquefies and the berries turn brown. So in Missouri, this is a common problem for our vignoles, which is the most planted white grape in Missouri. This is due to their tight clusters and thin skin, and especially happens when we have heavy rain events. This is also prevalent in other cultivars such as Chardonnay, Traminette, Vidal Blanc, and Chambourcin, which make up a considerable amount of grapes here in Missouri. The sour rot disease complex has 3 associated organisms. This is yeast, acetic acid bacteria, and Drosophila fruit flies are necessary for this disease. Another important factor are wounds and humidity. Berry damage can happen through multiple ways throughout the season, such as bird damage, as you can see on the left. Also, separation from the pedicel, which happens when these grapes grow very close together and become compact. Insects can also wound the grapes, as well as hail events, and most importantly, rain, which happens a lot in Missouri towards harvest, where we have heavy rains, which leads to the swelling of the berries and then berry splitting as these thin skin and tight cluster varieties become affected. Wounds are important for sour rot as grapes accumulate sugar when they ripen. These wounds expose that sugary carbon source for yeast and bacteria, which increase into the immediate area. These wounds make an aerobic environment, which converts ethanol to acetic acid, and the fruit flies are attracted to the volatiles in overripe grapes that contain these fermenting yeast and acetic acid. So female fruit flies prefer wound sites to lay their eggs. The yeast and bacteria are diverse and make up the natural microbiota of the grape. The microbial populations increase as berries ripen, and it's not only isolated from the surface of the berries, but also within the berry pulp. Yeast and bacteria are present before sour rot begins, and these populations increase when berries become damaged and affected by sour rot. So in sour rot, we have a microbial interaction. Yeast growth leads to early fermentation on the vine, converting that sugar to ethanol. And along with that, we have acetic acid bacteria populations that grow and metabolize that ethanol to acetic acid. Fruit flies are responsible for the spread of sour rot associated microorganisms that accelerate this fermentation process. And a high microbial load and acetic acid accumulation is detrimental to our wine quality. So the main characteristics of sour rot are the browning of berries, the liquefaction of the berry pulp as it kind of oozes throughout the cluster, and the strong smell of vinegar, and fruit flies are always present in the vineyard. So they're flying around as adults, mating, laying eggs, and you can see pupae on the outside of the clusters. Even when you open up the clusters, take your pruning shears, you can see the pupae within them. And also you can see larvae that feed within the berries. So in the vineyard, when we see a high amount of sour rot, you also see a lot of fruit flies that conglomerate around these clusters. And during this time, you can almost breathe them in just because there's so many present sometimes. And multiple species can exist within one cluster as they lay eggs and feed within these sites. And as you can see over on this right side, the larvae are present within the clusters. And these larvae feed on the grapes as they develop and then mature into adults. So they are also very important for sour rot formation. Fruit flies are necessary for sour rot, and this can include a lot of different Drosophila species. The main species would be Drosophila melanogaster, which is attracted to decomposing fruit and take advantage of damaged fruit. Other species are also important, such as Drosophila suzukii, Drosophila similans, and many others. However, Drosophila melanogaster are mainly found on sour rot clusters that are experiencing severe symptoms late in the season, and they prefer these damaged berries. So in a general sense, here we're looking at a life cycle of Drosophila suzukii, which is also common for any Drosophila species. We have the egg that hatches within 12 to 72 hours, and then this develops into a larva, which has 3 different life stages. And so this takes about 5 to 7 days to complete their 3 different life stages. The third larval instar will then pupate and then develop into an adult within 4 to 15 days after the egg was first laid. And so the new fruit fly adult can live 20 to 60 days over the season, and one female can oviposit about 3 eggs per site and has about 350 eggs laid within her lifespan. So in a vineyard, we have a very fast generation of life cycles. This can occur in as little as 10 days sometimes. So there's a lot of overlapping generations, which can snowball out of control. Fruit flies have symbionts, such as yeast and acetic acid bacteria, that are present on the surface and within the guts of fruit flies. So this is obtained from the fruits, and the adults are attracted to fermenting yeast and acetic acid for their sites that they like to lay eggs. This is present in the gut to help digestion and immune system, which is why they have a yeast diet. So where do the fruit flies come from? They overwinter in habitats that protect them from freezing temperatures, such as leaf litter and man-made structures. They also come from field edges. So this could be vegetation that is not treated by insecticide, as well as forest habitat that support fruit fly populations when the crop hosts are unavailable during the winter. And so during the season, Drosophila pressure is usually higher along the forest edge and field habitat that contain these alternate hosts. And during the winter, populations can develop winter morphs that are resistant to freezing temperatures, and some even enter diapause and survive on plant material during the cold winter months. And for sour rot management, it is important to rotate modes of action as much as possible to avoid building resistant fruit fly populations, since we have so many generations occurring during the season. There are very few products available that are inexpensive and have a short pre-harvest interval. So here's an example of some of them that are commonly used to treat fruit flies. And it's very important that we want to save these chemistries just because they have a short pre-harvest interval and because they are relatively inexpensive for us to use. Resistant fruit flies have been found in the Finger Lakes in New York. These populations are heavily sprayed with these 3 compounds, zeta-cypermethrin, acetamiprid, as well as malathion, over a course of seasons. And so this is comparing a wild type fly with ones collected in the vineyard. And as you can see, we have a lethal concentration that does not work on any of the field collected flies. And this is especially the case for zeta-cypermethrin and acetamiprid, where we have 4 times the lethal concentration, where we have around 60 to 80% of the flies surviving that. Our understanding of sour rot and the causal agents have only recently been determined. We are unsure of the exact contribution of the fruit flies. We just kind of know that they are causal for this disease. And their uncertainty of the disease has limited the development of reliable management practices for sour rot. So Drosophila fruit flies with the microorganisms, they act together to cause disease. And controlling the fruit flies is the most important step in reducing sour rot when we have fruit flies, yeast, acetic acid bacteria, and also wound sites on the grapes. Previous spray trials have looked at pre-harvest applications of antimicrobial agents and insecticides alone and in combination. Based on previous research, antimicrobials alone do not reduce sour rot. Here we're looking at no insecticide treatments, which is the white bars, and treatments that include an insecticide, which are the black bars. And as you can see, all the treatments that included an insecticide lowered clustered area of disease. So insecticide is important for controlling sour rot, and antimicrobials alone do not reduce sour rot. And they found less control in treatments that did not receive an insecticide. Further expanding on this, we see that pre-symptomatic sprays are more effective. So these sprays were initiated before sour rot symptoms were visible. And we see these are various different antimicrobial agents. When Acetate 2.0 was applied pre-symptoms, we had a lower disease of clusters when we use an insecticide in combination, as well as various antimicrobial treatments pre and post symptoms. Previous spray trials have shown that antimicrobial and insecticide sprays applied before symptoms provide the best control of sour rot, and insecticides significantly reduce the number of fruit flies recovered from these berries. Antimicrobials alone do not reduce sour rot, and insecticide applications are more critical as they target adult fruit flies. So sour rot is managed in Missouri using a current industry practice of weekly sprays of insecticide and antimicrobial cocktail every week starting at 15 bricks until harvest, which can be 4 to 5 or even more sprays depending on the season. This does not account for the fruit fly populations or whether a disease is actually present. And so vineyards commonly wait for symptoms to develop before initiating applications due to the cost of these applications and uncertainty of when sour rot actually develops in their vineyard. Here we're looking at some of my graduate work, which we conducted a spray trial using Oxidate and Mustang Max mixed together, applied at full rows based on Brix measurements taken at each week. These treatments were rated at harvest for severity and incidence, and this was conducted in 2 vineyard sites of Vignoles in Missouri. The purpose of the spray trial was to determine efficacy of reduced sprays 2 to 3 applied at specific Brix measurements and compare this to weekly sprays initiated after 15 Brix, which included 4 to 5 sprays. Sour rot is rated based on severity, which is the percent of area of berries with sour rot, as well as incidence, which is the proportion of clusters with sour rot out of the vine. So this is infected over the total as a percentage. So in 2018, the treatment applications included 16 and 20 Brix for 2 total sprays, 16, 19, and 22 Brix for 3 total sprays. This was compared to our industry standard of sprays starting at 15 Brix for a total of 4 to 5 sprays, which we used as our control treatment because we couldn't ask vineyards to have a no spray control. We found that there was no significant difference between 2 and weekly sprays. So here we're looking at severity incidence as a percentage on the left, and this compared our 2, 3, and 4 to 5 sprays. As you can see, we have no significant difference between severity between any of the treatments, and we have no significant difference between the 16, 20 Brix as well as the 16, 19, and 22 Brix for the sour rot disease incidence. What was really interesting is we saw a significant difference in the 16, 19, 22 Brix, as well as the weekly starting at 15 Brix for sour rot incidence. So for the weekly, we actually saw a higher disease incidence. In 2019, we replicated the spray trial with the 3 treatment applications. However, we modified one of them to include 2 total sprays, one at an earlier Brix level. So this second treatment was at 13 and 18 Brix. And we compared these against the industry standard of weekly sprays. Here in this year, we found that there was no significant difference between any of the treatments, between severity or incidence. So for our 1620 and 1318 Brix, this was not different than spraying 4 to 5 times starting at 15 Brix. And what was really interesting is we saw in the 13 and 18 Brix treatments, we saw a slight reduction in the amount of sour rot severity and incidence, which kind of hinted at the fact that if we start managing sour rot earlier in the year, then we can have a decrease in disease incidence and severity. However, this was not significant in our observation. We basically found that there were more sprays did not equal better control, and reducing these sprays did not differ from industry standard when applied after symptom development. So this is after 15 Brix. And spraying 4 to 5 times to control fruit flies does not offer more disease control, as we saw similar severity and incidence for 2 sprays. The implications of the spray trial, we found that less sprays are less costly both financially and environmentally. This can prevent resistant Drosophila populations from developing in the vineyards. And this research demonstrated the need for a better understanding of when symptoms develop so that chemical management strategies can be adjusted, as sour rot symptoms may actually begin before 15 Brix. And another question we wanted to ask is what other insecticides can we use to manage sour rot? In this spray trial, we tested the efficacy of using neem, which is azadirachtin as an active ingredient, which is extracted from the neem tree seeds. This is a natural occurring pesticide and is OMRI approved and reduces insect feeding and acts as a repellent. This also disrupts insect molting, which inhibits the hormone ecdysone, which is critical for larval molting. And this is in available products as Azagard and Azadirect. In our neem trial, we used a combination of antimicrobials as well as Mustang Max mixed with neem, which is azadirachtin. This was applied in full rows based on our 15 Brix application, which is conducted weekly. And this is rated at harvest for severity and incidence, similar to other spray trial, using the same 2 vineyard sites of Vignoles in Missouri. In our neem trial, we had weekly applications starting at 15 Brix of an antimicrobial mixed in with Mustang Max, mixed with neem, which is azadirachtin, as well as another treatment that included antimicrobial plus neem. And then we had a treatment of neem alone. And in vineyard one, we're looking at severity and incidence on the left, and this vineyard had a high amount of sour rot and was heavily impacted this year. We found that between the Mustang Max and azadirachtin and antimicrobial cocktail, that we did not see a significant difference between those three chemicals when comparing to azadirachtin alone over on the right. And this is the same case for incidence and severity. What was really interesting is we saw that azadirachtin mixed with an antimicrobial agent actually increased incidence significantly between any of the treatments, and as well as increased severity significantly when comparing Mustang Max with azadirachtin and antimicrobial agents. This was replicated in our second vineyard site, and as you can see on the left side, we had slightly less sour rot in this vineyard. So we've also found that there was no significant difference between incidence between any of the three treatments. However, we did see, again, a slight increase in incidence as well as a significant increase of severity in azadirachtin and antimicrobial agent treatment. In this vineyard, we also had a similar result. When comparing Mustang Max, azadirachtin, and antimicrobials together, we did not have a significant difference between incidence or severity when comparing to just azadirachtin alone. In a spray trial, we found that there was no significant difference in sour rot when comparing Mustang Max, antimicrobials, and azadirachtin with azadirachtin alone. So basically, one compound, azadirachtin, was just as effective as three compounds, which was our Mustang Max, antimicrobial, and azadirachtin. So this implies a cost-saving benefit, as well as another compound to use in rotation. What was really interesting is that azadirachtin with an antimicrobial, which in this case was Oxidate 2.0 and Spore Quell, resulted in slightly higher sour rot incidence and severity for both vineyards, which indicates that there's a possible interaction between these two chemistries. And these results were consistent for both vineyard sites, one with high disease severity and incidence, as well as the other vineyard, which had a relatively low disease severity and incidence. The most important step in management is to stay ahead of the fruit flies and to scout for fruit flies and sour rot on a weekly basis. So it's important to look for damaged grapes, which can happen by birds or berry splitting as we have extended rains and humidity. It is important for vineyards to incorporate monitoring traps in the vineyard to determine fruit fly presence for early detection. Making monitoring traps are relatively simple. Here we're looking at an example of a monitoring trap created by Cornell University by Carroll in 2014. Here we have a red solo cup hanging in a canopy that has dish soap with apple cider vinegar, as well as a yeast dough within the sample cup. And so this is a bait and drowning solution with a volatile attractants of yeast and apple cider vinegar. And so this will attract all types of Drosophila species. I've seen even simpler monitoring traps used in a vineyard, where we just have apple cider vinegar mixed with dish soap inside deli cups that are placed on the outside of rows. These deli cups have drilled holes in the side and are checked on a weekly basis to look for fruit flies, and the solution is mixed out. And then they are removed later in the season as we have fruit flies accumulating. An important question for vineyard managers ask is, what is the source of fruit flies? So to have a constant question of when do the fruit flies show up, and think about their neighboring fields as well as forest or other field habitat that they could be coming from. What is also important is to consider that they are actually coming from within the vineyard and surviving over winter there. Another important consideration is the level of immigration from the outside of vineyard. So weekly sprays may actually not be effective if they're coming from an outside source if you're next to other fruit crops as well. And so the constant question that we are asking and still needs to be researched is, when do fruit flies enter the vineyard? It is important for you to scout weekly after veraison by using fruit fly monitoring cups, as well as looking for the presence of sour rot berries. So through disease scouting and fruit fly trapping, you can determine whether or not it is essential to spray early that season. And it's also important to focus on the weather, as heavy rains and high humidity can lead to heavy sour rot infestations as the canopies stay wet. In sour rot management, it is important to delay heavy fruit fly pressure and monitor fruit flies and disease on a weekly basis. So in spraying insecticide early, before 15 bricks, if fruit flies and sour rot are present, will actually control early fruit flies and prevent high fruit fly numbers by the time of harvest. Another important consideration is canopy management and improving the microclimate by increasing sun exposure and promoting air movement through the canopy. So this allows the clusters to dry when we have heavy wet periods. It is also an important factor to consider harvesting early before sour rot affects the majority of clusters. So when we have high sour rot severity and incidence, this leads to acetic acid accumulation, which has a legal threshold of 1.2 grams per liter in wine. And also, sour rot significantly reduces the extractable juice up to about 45% when these berries become dehydrated and the pulp essentially liquefies out of the cluster. With that, I would like to acknowledge the Missouri Wine and Grape Board Research Committee grant for funding my research, as well as the Milliken Endowment Fund. And I would like to provide thanks to the vineyard sites that allowed me to sample, as well as thanks to my main advisor, Dr. Megan Hall, and my committee members, as well as our lab techs, Ziwei Fang and Connie Liu. And with that, I would like to thank you for listening to my presentation. And if you have any more questions, feel free to contact me at my email provided. Thank you.